Buenos días, muchas gracias a todos por venir y bienvenidos. Que tengan un excelente día y nosotros venimos de Uniparts en colaboración con IDT. Uniparts es el distribuidor de la marca en México y venimos con una serie de ponencias titulado De la edición genómica a la secuenciación, con la idea de que ustedes se lleven una introducción a los portafolios de IDT, pero sobre todo que tengan la puerta abierta de la mano de expertos en cada uno de estos portafolios para que se puedan asesorar en aquellos protocolos que quieran implementar, algún flujo de trabajo que quieran optimizar y que sepan que tienen el servicio de primera mano de ellos. Con esto damos la bienvenida a Branimir Burgarja, PhD, especialista en edición genética mediante CRISPR y genómica funcional, con amplia experiencia apoyando a investigadores en la implementación de tecnologías para investigación básica, pero también en la parte translacional a desarrollo de terapias. Actualmente se desarrolla como científico de aplicaciones de CRISPR para IDT Estados Unidos, donde lidera el equipo de especialistas que involucran a soporte científico. Obtuvo su doctorado en biología molecular por la Universidad de Chicago y forma parte de IDT desde 2019. Entonces, demos la bienvenida al Dr. Branimir. Please, Bran, thank you for joining us. I'll let you begin. Thank you.
Yeah. Can everybody hear me? Okay. Thank you for the kind introduction, David. It's really good to be here. Thank you for the invitation. And my goal is to give you an overview of what we do at IDT, how we support genome editing, show you some of the tools, solutions we have available, and most importantly, to get questions from you so I can understand better what you're doing and see how I can help you move your research forward. So I've been with IDT for 15 years in 3-4 different functions, and I've been leading the functional genomics and CRISPR team in the Americas for the last 6-7 years. So I've had a chance to see all of IDT's solutions launch from research all the way to commercial. So it's been a really nice journey, really good experience. And if you have any questions about anything that I talk, this is intended to be more interactive. So feel free to raise your questions, interrupt me, stop me. I want to make sure that I answer any questions you may have. So a little bit about IDT first. So IDT is a global oligo manufacturer. Right? So we have our main facility in Coralville, Iowa, which is in the middle of the United States, maybe about 3.5 hours west of Chicago, where I live. That's where almost all of our oligos are made. That's also where we have research and development. So everything that you see here today was also developed at IDT. So what that means for you is that we don't just make the products, we also do the innovation. So we have a lot of know-how in terms of how to use, how to deploy these tools. So feel free to rely on us, not just for the tools, the reagents, but also the protocols and the implementation. We do that all at IDT. But just to highlight, we are a global company. We make more oligos every day than any other company in the world. We have facilities in San Diego, North Carolina, Singapore, Belgium, so we have really a global presence. For the most part, everything that you would get here in Mexico would be made in Iowa. And here's just kind of high-level numbers. We have a lot of customers. This slide is a year or two old, so there's probably a lot more than this. We make a lot of oligos every day, more than 120,000 today. Everything is high throughput. We make 96 oligos at a time in machines, warehouses of machines that make these oligos 24 hours a day, 7 days a week, every day of the year. And what a lot of our customers don't know is that the instruments that make oligos are made at IDT. So we have our own engineering group that does everything. So fully integrated from the machinery all the way to the R&D part. All right. So as I mentioned, we make a lot of oligos every day. And for most of IDT's history, people knew us as the oligo company. We make primers for their PCR reactions. But we also enabled a lot of other technologies to flourish as a result of our ability to make good custom oligos. So even to this day, at the heart of what we do is custom DNA and RNA. But over time, we have evolved to really transform our offerings to more packaged genomic solutions with R&D that is accompanied with it. So what we'll focus on today is going to be mostly next generation sequencing, which you hear from my colleague Rachel. I'll focus mostly on CRISPR. This has been one of the key areas for IDT as the technology launched in 2012. We really latched on because we have an edge in being able to support this type of research. But we also do PCR, basic PCR, qPCR. We have a lot of reagents in that area. You'll hear a lot about that from Riley, as well as our synthetic biology group, which is, again, taking these oligos, repackaging them into double-stranded fragments, genes that can enable you to do all sorts of genomic assemblies and any type of synthetic biology work. Okay, so this is a little bit about IDT's CRISPR innovation journey, just to give you a high-level overview of what we've done. I can't go into detail on everything here today, but I'll show you some of the main highlights. Really, I think everything starts with our ability to make oligos, right? In order to provide synthetic CRISPR reagents, you have to make really good quality RNA. So we've been on this journey since the first days of IDT more than 35 years ago. But at the dawn of the CRISPR technologies, which really launched around 2014 when the first papers came out, we really jumped on it very quickly. And we enabled synthetic reagents to be ordered almost within 6 months of that paper. So we started out by selling cRNA trace RNA complex because it was the most cost-effective and easiest way to enable researchers to get synthetic reagents. But as you can see here, we've been on a long journey, optimizing new systems like Cas12a, improving enzymes so they work much more efficiently, being able to make longer RNAs like the sgRNAs, improving donor templates, an entire suite of solutions for doing precise genome engineering, which I'll show you in a little bit, all the way to advanced analysis systems. And in 2025 and 2026, we've actually enabled therapeutic applications of IDT's guide RNA. So we can now make cGMP guide RNAs. And we also offer off-target analysis services, which is part of the safety packages that are required if you want to submit a therapeutic to the FDA. So we've done a long journey and we continue to innovate. So as we speak today, this is a high-level overview of our CRISPR portfolio in terms of the solutions and tools we have available. And we like to break it into 4 columns here. So we have the design column, the reagents column, the HDR column, and the analysis piece. This is basically describing the workflow. There's one column that's not here. I actually don't have a slide for it, but feel free to ask me about it, which is the delivery part. How do we get these reagents into the cells or tissues? Not something that we have tools for, but we have a lot of expertise, a lot of protocols, so feel free to ask me about it. But on the design side, we have tools that can help you design the guide RNAs. So we can design guides, whether it's one gene at a time, hundreds of genes at a time, lots of different formats you can design, from one guide targeting an exon to multiple guides targeting an exon. All depends on what your strategy is. We have tools that can help you design the HDR experiment, so both the guide RNA as well as the donor templates. We have all the reagents that are most commonly used, so everything on the Cas9 side, the enzyme, the guide RNAs, on a Cas12a side, which is another system we support, a lot of reagents there, as well as anything custom that you want to build, we can also enable that as well. We have various types of donor templates available, all optimized for more efficient and higher HDR rates, which I'll go into a little bit, as well as new innovative tools such as our HDR enhancer, which can further boost your knock-in efficiencies. And I'll spend some time with that as well. And then we have been focusing a lot lately on the analysis part. So how to actually make sense of all the experimental data that you just generated, or actually how to assess the editing outcomes, right? So it's easy to order the reagents, it's easy to generally deliver them into cells. It's more challenging to make sense of what the editing actually is and how it worked out. So we have a lot of tools available for that as well. All right, so before I go deeper into our tools and offerings, I'd like to maybe start off quickly by giving you a quick intro into what CRISPR really is and how most researchers do it, and then how we fit into each part of the workflow. So at IDT, for the most part, we work with 2 main systems. One is based off of SBCas9, And then we have tools available under CPF1, or what's now called Cas12a. And we have two different enzymes, AsCas12a and LB, depending on your application. And in both cases, Cas9 as well as Cas12a are what I would call RNA-guided endonucleases. So they're programmable RNA-guided endonucleases, which for you, what that means is you can load a Cas9 with a specific guide RNA and target a specific region of the genome to create a double-stranded break. That's all it does. And what then happens is, depends on what you want to do. So you can either exploit this double-stranded break to do allow the cell to repair through non-homologous end joining, which means that you're trying to create indels or insertion deletions that can throw your gene out of frame. And that's usually used to generate gene knockouts. So if you want to study biology as a result of removing a gene from the system, very useful. That's the most common application of CRISPR-Cas9. On the other hand, you can also take advantage of these double-stranded breaks to do very targeted genome engineering. As an example, you find a mutation in a patient, you want to bring it back into a cell line and study how that mutation impacts biology. For that, you would need to also introduce a donor template. And this is a little more challenging. It's a little bit harder to execute. It requires more design expertise. And in terms of efficiency, it's also more challenging than just doing a knockout. So if you wanted to change alleles, mimic these tags, like adding a GFP tag, or maybe like a flag tag or a MIC tag, all those can be achieved using HDR. And I'll show you some of the tools from design as well as the reagents that can enable you to do that more efficiently. But this is essentially how the vast majority of researchers use the tools that we have. Now, there's a lot of different ways that you can potentially bring the machinery into a cell. So when you think of CRISPR-Cas9, you need the protein and you need a guide RNA. But the protein can be delivered as a plasmid, where you can have the plasmid express both the Cas9 as well as the guide RNA. You can potentially deliver the protein as mRNA, and the guide RNA can be delivered as a synthetic DNA fragment, in vitro transcribed guide RNA, or synthetic guide RNA. Or you can potentially deliver ribonuclear protein, where you have purified protein that you couple with a synthetic guide and you give that in. So this is an example of, I would say, a long period of work at IDT testing different formats, but just to give you a sense of what we do and why we do it. So when you think about plasmid delivery, so you have a combined expression plasmid where the Cas9 and the guide RNA are on the same DNA. You transfect it into cell. That plasmid has to go into the nucleus. It has to be transcribed, exported out of the nucleus, translated, and the plasmid sits around for a long period of time. And you can see here when you look at, this is a Western blot of Cas9, you start very slow, when you transfect the plasmid. It takes maybe 4 to 8 hours to start seeing some protein, but then by 72 hours, there's just an overabundance of protein. It begins to just create more and more and more protein. So this is good because it gives you very high on-target potency. It'll be very efficient at the on-target. However, it's bad because it sticks around too long, which means it'll cut at the intended target, but also has a lot more opportunity to cut at off-target. And what we generally see is that sustained expression of these reagents creates a lot more off-target editing. In contrast, when you do ribonuclear protein delivery, you have a reverse image of the Western blot, right? You have a high amount of Cas9, so you transfect RMP into your cells, it goes in, it goes directly into the nucleus and creates a double-stranded break. And over time, protein gets turned over. And you can see here by 72 hours, most of the protein is gone. So we like this approach because it's fast on and off. Ideally, when you do genome editing, you want to edit the site you want, and you want the reagents to go away as quickly as you can so you can limit off-target editing. So at IDT, for many reasons, including ease of use, the experimental outcomes, we have honed in on RMP as the preferred method for the vast majority of applications. Not for everything. There are some applications where you still may want to think about using expression plasmids or using mRNA, but for general applications, doing genome knockout, genome knock-in in cell lines, RMP is the way to go. So this is a high-level overview of the proteins we have available at IDT. So as I mentioned, we mainly work with Cas9 and Cas12a. And with Cas9, we have multiple different flavors. So on the left-hand side, you'll see wild type and hi-fi. So those are, I would say, the most generally used enzymes. The wild type enzyme has the highest potency. So this is an enzyme that we engineered at IDT by changing the NLS composition, so the nuclear localization signal. We changed the number of NLSs, the position on the protein, the linkers. So everything has been optimized to continue to give you improved editing efficiency. So if all you're interested in is the highest editing efficiency, the wild type enzyme is the way to go. If you're looking at an enzyme that can give you a good compromise, so it has good on-target potency, but can vastly reduce the off-target, then the hi-fi enzyme is the way to go. These other are available for more, I would say, niche applications, more boutique applications. So you can use this to enrich for cells if you want to, right? It's only useful if your delivery is not very good. So if you have very robust delivery, enriching is not really going to do anything. But if your delivery is subpar, let's say 20, 30%, then having a way to pull out the cells that are successfully transfected can be very advantageous. And then we also have Cas12a. I would focus in here on the ultra enzymes. Those are the ones that we re-engineer to have higher activity, higher potency, and they're really on par with Cas9. The main reason why we have these two systems in the first place is down here. So what this means for you is that with Cas9, you can target using an NGG PAM. So pretty much every 8 nucleotides in a human genome has an NGG, right? So it gives you a lot of opportunity to easily target genes of interest. Cas12a is a little bit more restrictive in terms of PAM availability, but it can be useful in certain situations where Cas9 is not the option. So I generally find that 1 out of 10 researchers will use Cas12a for any number of applications, whether it's gene knockout or, as you'll see in a little bit, gene knock-in requires very precise cutting, very close to the insert that you want to generate. So being able to select PAMs and enzymes that can allow you to do that can be advantageous. So it comes in very handy in those applications. As I mentioned, the Cas12a PAM is more restrictive, so it's harder to find targets. But because it's more restrictive, it's also it has a lower off-target profile. So some researchers prefer to use this enzyme because of that. Okay. And just to demonstrate some of this innovation, as I mentioned, we do a lot of work at IDT to innovate these tools, right? So we have a protein engineering group that evolves enzymes. And not only do they evolve enzymes, they also engineer them. So there's a protein evolution part where we are trying to find new mutants with properties that are of interest to us. So as you can think of for Cas9, we want the enzyme to be very active, but we want to produce mutants that reduce the off-target editing, right? For Cas12a, the wild-type enzyme was not very potent, right? So we wanted to engineer mutants that increase the potency. All that is done with the same group that engineers the enzyme, but also creates novel mutants. And the engineering part, as I mentioned earlier, is introducing NLSs at different positions. So we iterate everything. Do you need 1? Do you need 2? Do you need 3? What kinds of NLS sequences can we test? Do we want 1 on C terminal, 1 on N terminal? You can then change the linkers between the NLS and the protein. All those things take a long time to iterate until we find something that gives us an increase in editing or other features that we're looking for. But these are just 2 examples I think are really good innovation at IDT around the enzyme. So as I mentioned earlier, hi-fi enzyme is probably one of the blockbuster enzymes that we launched at IDT. So this is a novel mutant that essentially maintains activity on par with the wild-type enzyme, but reduces the off-target. And we actually did this in collaboration with a group at Stanford University where they're looking for, they had a target that they wanted to use to treat sickle cell. But that particular guide had a very nasty off-target site that just was very difficult for them to get rid of. They used our enzyme, the hi-fi Cas9, to really lower that off-target profile down to a level where it's acceptable as a therapeutic. And this enzyme to this day is being used by multiple groups in clinical trials. So if you want to read more about it, how it was developed, the engineering screen, the outcomes, you're more than welcome to look at this paper. And likewise, as I mentioned for Cas12a, there's a company called Editas that owns a lot of the IP around Cas12a, and they use this in their clinical development. But the wild type enzyme, as I mentioned, is not very potent. So they actually collaborated with IDT to develop this novel mutant that actually now has therapeutic use, whereas the wild type enzyme was not very useful, then the new mutant is. And we make all this available both as an REO enzyme and then for them as they needed for their therapeutic applications. And just to demonstrate how the work with HiFi kind of looks, this is just an enzyme. I mean, this is a site that we commonly use in the gene EMX1, which has a very good on-target site, but also has a lot of off-target activity, as you can see here. And it's an example of how much the wild type and the HiFi can sometimes differ, and really where the benefit of the HiFi enzyme comes in, where you can really clean up all the off-targets that are at this location, and you maintain on-target potency that's on par. We like to say that for about 90 to 95% of the sites, the on-target potency between the HiFi enzyme and the wild type is going to be very comparable. There is a small number of sites where it can take a hit in activity, but it's very unusual. And this is the work that we did with Editas. And again, these are very clinically relevant cell types, clinically relevant targets for them. And they did a comparison in the paper that I just showed, comparing a lot of different targets across multiple cell types, comparing the wild type Cas12a versus the ultra mutants, and you can see there's just a massive difference. So at the end of the day, the message is you have two systems that work comparably. If you need to work with Cas12a, very active system. If you need to work with Cas9 and you're thinking about potency, we have the wild type enzyme. If you're concerned about off target, we have the HiFi enzyme. So when it comes to guide RNAs for Cas9, there are two different options you can potentially work with. As I mentioned earlier, our first product at IDT was actually a two-part guide RNA, and it made a lot of sense both from the synthesis part as well as the economic side. Here you have a short cRNA, so this is 36 nucleotides, that's coupled to a universal trace RNA. So you could, as a researcher, order a trace RNA, have it in the lab, and anytime you need to target a new site, order a short cRNA. Easy to make, easy to ship, cost-effective. And this is still something that we offer to this day. It's very good for basic applications, RNP delivery, cell lines, all that works quite well with the two-part system. On the right-hand side, as the technology has evolved, as synthesis got better, as volume increased and the cost went down, we actually started making this as one piece called the single guide RNA. And this is, I would say, where the vast majority of researchers are shifting to single guide RNAs because it's easier to use. You don't have to anneal to oligos. And then for primary and more difficult conditions, technically the single guide RNA is more stable. So it tends to work better if you're working with challenging cell types. And the reason for that is that this two-part system here has 4 ends, right? So the exonucleases can degrade much faster than they can here. So in this case, especially if you're working with mRNA, for example, for any in vivo applications, you definitely want to use sgRNA. Anything that's therapeutically relevant, generally everything defaults to one RNA oligo because it makes it a lot more easier to qualify and submit QC packages for your regulatory bodies. And again, this is just a high-level summary of the different options we have for the guide RNAs. So there's the Cas9 system with 2 different two-part options. It all depends really on the application you're working with and how much stability you're after. So for most basic applications, all of these will pretty much work well. The two-part tends to be more economic because you're ordering a shorter cRNA with a universal trace RNA. But as you get into more challenging conditions, you definitely want to move into formats that are more stable, both from being a single piece as well as more chemically stabilized. And then we also offer Cas12a options. These are basically just short cRNAs with end modifications that are suitable for the vast majority of everyday applications. If you need anything outside of this, if you work with a system that's not Cas9, Cas12a, Cas13, prime editing, any type of unique modifications you want to introduce into guide RNA, we can also make those. We have the tools, as I mentioned. So we really support customers from design all the way to analysis. So when you want to do genome editing, you need some way to design the guide RNAs, right? So we have a tool available on the website that's free for you to use, very user-friendly, where you can put in your species of interest. So it has human, mouse, rat, zebrafish, C. elegans already pre-populated. You can put in your gene symbol, the number of designs, and the tool will just provide an output where you'll have exons in green, introns in black, and it'll show you the locations of the guide RNAs, and it'll give you a scoring output, kind of telling you what the likelihood of this guide RNA being more potent versus less potent, and it'll show you the off-target editing or the off-target potential sites that are there that you can also evaluate. If you have something that's not human, mouse, rat, zebrafish, C. elegans, we also have a custom design feature where you can just put in your custom sequence in FASTA format, and it'll design guide RNAs against that sequence as well. And then if you have sequence that you design somewhere else from a different tool, we'll also have a way for you to check the potency of that or the score, the on and off target of that sequence using our design checker. So it's very simple, very user-friendly, but it allows you to create a design, and from design, you can then order those reagents as well. This is just an output of what the tool actually looks like. So this is a design generated. It'll show you the protospacer, the FAM sequence, the on and off target scores, and then you can open up this little tab here. It'll show you all the potential off-target sites that we think are feasible. Every red here is a mismatch. Every highlighted blue is a deletion. So you can see exactly where they are, what the chromosomal location of the off-targets are, and what the location, if there are any, in any known genes. So you can go down and see which guide RNAs might be most suitable to you. Just for reference, our scoring goes from 0 to 100. So 100 is the best for both on and off target. And keep in mind, this is all in silico predictions. So it's giving you an enriched set of sequences that we think are going to be more potent versus less potent. And then you can use both the on and off target to kind of rank what's more important to you. If you do anything more high throughput, we have a CRISPR libraries design tool. So the tool that I just showed is 1 or 2 genes at a time. Kind of makes it easy to do everyday genome editing. But if you're doing high throughput sequencing, high throughput array screening, you can put in 20,000 genes into this tool. It'll create all the designs for you. And it has a lot of built-in flexibility in terms of you can dictate how far the guide RNAs are from each other. This tool can do pool by gene designs where you can design multiple guides to target a single exon, and will provide those sequences all in a single tube or in a single well. And the tool also will design the guide RNAs in such a way that it'll force predictable deletions that create frame shifts. So it generates higher likelihood of having a functional knockout. So this is for anybody that's doing anything more high throughput. And you can, as I mentioned, you can specify the location or the distance of the guide RNAs. As I'll show you in a little bit, you can also ask the tool to design complementary primers that can span where the guide RNAs are. So you can build amplicons that allow you to then sequence and analyze by NGS the editing efficiencies. And you can dictate any number of sequences per gene. If you need more than this, we can also do that, but generally up to 4 is good enough here. And this is just a quick overview of what the customization looks like. So if you wanted to order 100 genes in a plate, you can customize it any way you want, from the plate type, the type of product, the PCR plates, whether you want PCR, deep well, echo plates, anything that you can think of, we can probably do. And if it's not here, just ask us. Dry, wet, concentrated at your desired specs. All that can be done. As your research advances from basic RUO screening into something that's more translational, or if you're thinking about taking a particular target into the clinic, generally the quality of the reagents begins to change, right? So that you're introducing tools and reagents that are potentially going to mimic something that in the future might be therapeutic. Just a high-level overview that we can provide also purified guide RNAs, which means that we'll take the sequence that you normally order, we'll put them through HPLC purification to give you a product at the end of the day that has a higher full-length percent. What that means for you, cleaner sequences, something that's going to mimic that sort of future cGMP state. And in many cases, depending on the application, you also get a boost in editing. One area where this is going to be really important is if you're using mRNA. We currently don't have mRNA at IDT for Cas9. It's coming out September 14th. So in 2 weeks, we'll have mRNA available. So anybody that's doing in vivo editing, you wouldn't use RNP delivery. You probably wouldn't use plasma. You would probably, for something like this, use mRNA that you would package into an LNP, for example. And then with that, you would want the best quality guide RNA. So these would generally be more highly modified guide RNAs that are purified, that are then complex with the mRNA for that type of in vivo work. So that's going to be available soon. And this is just sort of a prelude to if you are doing something like that, we have that available as well. And this is just a UPLC chromatogram just to demonstrate that we make really good quality desalt sgRNAs. They're actually quite good, but you can get much better here if you actually purify BHPLC. For anything that's more therapeutic or relevant, generally you're looking for a purification or percent purity that is going to be more than 80%. So we generally guarantee that with our products. And again, this is the entire journey that our customers generally go through if they're actually thinking about the end outcome being more therapeutic. You start out more broad, you screen in the REO phase, small scale quantities. As your research becomes more translatable, right? You're looking to carry something that potentially from the bench into preclinical. Then generally the quality and the amount of the guide RNA changes. We can support that with HPLC purified guide RNAs, as well as much bigger yields if need be for larger number of cells that you edit, in vivo type of applications. All that can be supported. And then at the end of the day, we also have the ability to make cGMP reagents that are qualified for therapeutic applications. We also have a sister company that we work with, Aldevron, which you may not have heard of, but they enable us to really provide the full suite of solutions from REO to GMP for both the enzyme as well as the guide RNA. So IDT, as I mentioned, we support the REO space. We can also support the guide RNA in a GMP commercial phase if need be. But when it comes to enzymes, our sister company works with us very closely to enable our customers to get access to both the protein as well as mRNA that is required for any sort of therapeutic application. So we have the ability to support everything from one end to the other. All right, so I'm going to pause here for a second. Any questions? Okay. All right, so as I mentioned at the beginning, there are two main routes that our researchers use CRISPR-Cas9. So keep in mind, these are just scissors that can really precisely cut genomic DNA. That's, for the most part, what they do. And then on the left-hand side, as I mentioned, doing gene destruction. On the right-hand side is where I really like CRISPR. It's doing gene construction. Right? So you're really trying to create something that's important to you. It's genome engineering. It's really introducing things in that you can control. And you can use this for many different applications. Some researchers actually use HDR to do very precise knockout, where they want to knock in a stop codon. You can do a lot of things, right? But for the most part, I run into researchers that are trying to introduce SNP mutations that are more clinically relevant, or maybe introducing tags that allow them to track the protein, maybe a GFP tag, maybe a high bit tag, any number of things. So I'll show you some of the tools we have available. And if you have any questions, more than welcome to help with any project designs. I do that on a regular basis. So for HDR reagents, we have two types of dedicated donors. So as I mentioned, you create a double-stranded break, but then you need to provide the cell a donor template, which is the sequence you want to bring in. And the donor template has two features. It has your sequence and then homology arms, which are sequences that are identical to either side of the cut site. So we have two types of donor templates that we develop. One is HDR donor oligos, These are single-stranded DNA oligos. They're stabilized on both ends with phosphorate-thioate linkages and then end blocking modifications. And the reason we do that is we want to protect the oligo from exonuclease digestion. This is an ideal product for insertions up to 120 nucleotides. So anything SNP mutations, you want to introduce a restriction site, a high bit tag, MIC tag, flag tags, all those things can be done with this kind of product. They're easy to use, very effective, and something that we enable through our design tool, as you've seen a little bit. If your payload, if your insertion becomes bigger than 120 nucleotides, then the format of the template that you use changes from a single-stranded DNA to double-stranded. At IDT, we work with what are called HDR donor blocks. So these are double-stranded linear DNA fragments. And we have engineered them with end modifications that reduce blunt integration. So we basically don't want these to go in through homology-independent integration. We want it to go through HDR. And so that's one of the main concerns, problems with using linearized DNA is that it can also pop into the cut site through just homology-independent integration. So this is a tool set that we have for the donor templates. We also have HDR enhancers that we built. So HDR enhancer V2 is a small molecule that blocks NHEJ. I won't go into too much detail about that one. It works really well. It's something you add into cell culture media. So when you create a double-stranded break, you bring your cells to recovery media. That media contains small molecule. I'll talk a little bit about HDR enhancer protein. This is the latest innovation at IDT, where this is a small peptide that works directly to promote HDR. So it's a 53BP1 inhibitor. So it actually blocks the enzyme that blocks end resection and thereby promotes directly HDR. Okay. But I want to show you the tool, right? So HDR, as I mentioned, is technically more challenging to execute. The efficiencies are generally variable. There's lots of different things that go into making HDR work, but let's say efficiently or well. But we do have a tool that can help you to create designs. So to create the donor templates with the right features in them, as well as the guide RNA. So this tool is available on our website, free for you to use. It has really two areas that you need to provide details. One is input format, so you can put in your gene. You can select the species. If you have an accession number, you can do that as well. And then if you're just working with an enzyme that creates a double-stranded break, you can just click on wild type and hi-fi. It has a lot of other different features, which I won't go into details here. The vast majority of end users just work with a wild type or hi-fi enzyme. And when you click search, it takes you to a page like this, where you can select the specific edit location. So you can go into the chromosomal location with edit, click zoom, and it'll take you right to that location. And then we have what's called the mutation box here, or you can open up these two windows, and you can enter in whatever sequence you want. If you click on these amino acids, it'll open up a menu of codons. You can easily go and pick and choose what you want to work with. We have a pre-designed or pre-populated menu of tags, so HA tags in there, flag tag, 1X, 3X, everything that's more common is already pre-built with the different orientations you want to introduce them in. But the tool is incredibly easy to use, and it takes what I used to do manually. I can do this now in 2 minutes, right? I used to take half an hour to create a design because you want to build a donor template. You have to put in the silent mutations, all the features that you need to have in there, which now this tool can really make super easy to use. This is just an example of how the tools that we have available for HDR actually promote or can provide additional value to you, why they exist, right? And so here on the left-hand side, we're comparing our HDR donor oligos to unmodified oligos. So you can order like just a regular DNA oligo. You can order a DNA oligo that has phosphorythia modifications. And then the Altar modified oligos have both the phosphorythia modifications, as I mentioned. They also have end blocking modifications. And we can see that they provide a boost in HDR, right? So that's the reason why we decided to launch this product. They work better than unmodified or partially modified oligos. And then, as I mentioned earlier, this HDR enhancer V2, it blocks NHEJ. So when you create a double-stranded break, the first thing the cell wants to do is take broken chromosomes and bring them back together. And it does that very efficiently. And if that didn't happen, we wouldn't be here today. Every day, your chromosome gets broken by many different things: the sun, what you eat, natural repair systems. But it's very good at repairing these things. What happens is it repairs perfectly and it gets recut. And then at some point, it makes an error, right? And so that's the process that this is trying to block. And if you block that NHEJ repair process, it forces the cell to recombine with the donor template. And we see that it does a good job of boosting that. And so if you want to get the highest editing rates, in this case, a combination of these modified oligo plus this enhancer is a good idea. Keep in mind that when you block NHEJ, it's not the most gentle things on your cells, right? It can be a little bit toxic, so you have to make sure you do a good dose titration, the amount of time that you expose so that you get the best of everything. Healthy cells at the end of the day and an increase in HDRA. So it does take a little bit of optimization before you get to a place. But we have protocols that can help you start at the right place and make sure that then you optimize left and right in terms of how much exposure you want. And this is just to show you that these tools and these reagents, they work with either system. So for the vast majority of research with Cas9 is good enough, and the tool that we have is based on Cas9. But there are times where the specific SNP or mutation you need to bring in, there is no Cas9 guide, or maybe one that doesn't work, right? You try it and it doesn't cut. In which case, you have to go to option B. A really good option B is Cas12a. The tool does not support this, but I can help you. So I have the ability to design this, just laying down the sequence in a Word format, and we can pick and choose exactly where we can cut and where the insert needs to be. So just keep that in mind. And as I mentioned, the donor blocks that we have at IDT, the reason those exist is that we can actually demonstrate that we can get more perfect HDR and less off-target blunt integration by using that product. And so when you create a double-stranded break and you provide a double-stranded donor template, there are two ways the repair can go. On the left is what you want. So this is homology-dependent repair. The homology arm is combined with the genomic site. On the right-hand side are two different ways that this can go wrong, right? So at the on-target site, you can have just homology-independent integration, which means it just drops in without recombining. And you can usually see that very easily when you sequence, you see a duplication of homology arms. So it just went in. And then this can also happen at off-target sites, right? If it can happen on target, it can happen off target. And so here we use a mock off-target site. So basically it's a donor block that is not intended for that target. So it's an off-target site that cuts at 100% efficiency. And this is just looking at unmodified versus the HDR donor blocks. And we see a significant drop in the amount of blunt integration that we get there. So this is the main reason why we have this tool. And as a result of having less blunt integration, we get better HDR rates. We also have this protein that I mentioned, so the HDR enhancer protein. So this is innovation that we performed at IDT. So we're looking at a way to promote HDR that wasn't blocking NHEJ. NHEJ inhibitors do not have a path to the clinic. They're just too toxic. They're usually failed cancer drugs, and not something that I would ever want to have introduced into me. So we're looking for ways that can be more gentle, that doesn't cause any additional scarring, and something that potentially has a path to the therapeutic side. So we developed this, an engineered version of ubiquitin. So we call it I53. So this is a ubiquitin variant that has high affinity for 53BP1. And 53BP1 is an enzyme that blocks end resection. So when you create a double-stranded break, in order to get HDR to work, you have to end resect the ends, right? This enzyme blocks that. And so what we have here is a tool that works directly in HDR. By inhibiting this enzyme, we're actually promoting higher HDR rates. And this is an example. This is just high-level summary of what this protein actually does. So it can give you generally about a twofold increase in HDR rates. But the key here is that it does this with high precision. It's a protein. It's easily delivered into cells. It doesn't increase any off-target editing, doesn't increase the number of translocations, and something that can be easily added into workflows. So it's electroporated directly with the RMP in your donor template. And as I mentioned, something that we plan having available in a cGMP format soon. And here's some data we generated at IDT. So we're looking at a number of different target sites across 2 different cell types. So these are HEC293 cells, human iPSCs, and we see generally about a 2-fold increase in knocking efficiencies with the use of this HDR enhancer protein. But the key here is this is a very gentle reagent, easy to use, non-toxic, and very easy to implement in your workflows. All right, so this is my last section, and it's going to be around gene editing analysis. So basically, how do we make sense of what we actually just edited? How do you detect the editing? And what are some of the options? Lots of options. It all depends on what you're trying to achieve, how much information you need, and also what your budget is. And so here are 4 different ways that I have supported in the past researchers who are trying to assess whether they have editing, what kind of editing. At the top is a mismatch detection assay. So the way this works is when CRISPR creates a double-stranded break, you create insertions or deletions. And if you have a pool of cells, let's say you have 1 million cells that you transfect, the insertions and deletions are going to be variable. One cell might have 1 base pair deletion, another have 5, one allele might have 3, 4. So you have a diversity of indels. You PCR amplify around the target. You have an amplicon, and the amplicon contains all the different types of insertions, deletions. You denature this by heating to 95, and you let everything come down slowly. As you let things come down slowly, they don't find perfect matches. So there's mismatches, and they form small bubbles. This enzyme T7 recognizes DNA-DNA mismatches, and it cleaves at that site. So it's a very easy way to detect if you had editing. You can run PCR. In your unedited, you have 1 fragment. In your edited, as a result of your enzyme finding these DNA loops, it'll create a cut, and you can have 2 predictable fragments that are easy to see. Very easy to use. It's not quantitative. It tends to underrepresent editing because the enzyme doesn't accurately detect 1 base pair mismatches, but it's good enough if you just need to know whether it worked or not, right? And we have a kit for this, and you just need PCR primers, and then the kit can take you through everything that you want. Sanger sequencing is easy to implement. I think it works really well for the vast majority of applications. And you just need a couple of primers. You sequence the pool of cells, and you can use tools like ICE that can help you deconvolute all the chromatograms and give you an assessment of the editing efficiency. So I generally find that most researchers, basic applications, as long as your editing is robust, so Sanger sequencing has a limit of detection at about 5%, so if your editing is below that, probably not the best thing to use. But if it's below that, then probably not a good experiment, so I would just change the guide RNA. And then we have also ddPCR. So this is another way if anybody wants to do, if you haven't access to a droplet digital PCR instrument. So this is using highly quantitative technology. And the way that this works, sorry, just a second, is that you have qPCR probes. You have a probe that has a fluorophore and a quencher. Maybe you have a FAM fluorophore, something that's more like a green color, and then you have another probe that has hex, something that's more like yellow, orange. And these probes will then anneal to your target. Right? You have two probes, and one probe sits over the cut site, and the other one sits somewhere else where you don't have any cutting. And the idea is if you create a cut and you have insertions or deletions under the cut site, this probe is sensitive to 1 base pair mismatches, and it'll just fall off. So you have 1 signal versus 2 signal. If it's 2 signal, no editing, 1 signal, editing. Very quantitative, very easy to use, but it doesn't tell you anything about the underlying sequence. So Sanger tells you the sequence, and it's pretty quantifiable. If your rates of editing are higher than 5%, ddPCR is super quantifiable, but doesn't give you sequence information. And then the last part here that I'll talk about is using NGS, so amplicon enrichment, which is, I would say, and the best of the best, right? Because you're creating an amplicon, you're doing very high depth sequencing reads to get very accurate, very quantifiable editing efficiencies, and also you can get to see everything about the sequence in there. So we have a tool that we use at IDT. It's our RAM-seq CRISPR analysis system, and it's amplicon enrichment that's tied to an analysis tool. So let me just give you a little insight into that. So the way that this works at IDT is we use a technology called RNase H2 or RHPCR. RHPCR is using primers that have a blocking group on the end. So these primers cannot extend on their own. They have to be deblocked. The other feature in these enzymes is that there's a ribobase, RNA base in the enzyme. And this enzyme RNase H2 recognizes RNA DNA heteroduplexes. So you have primers that are blocked on the end, right, and have a ribobase. So essentially, without the RNase H2 enzyme, they cannot extend. In the presence of this enzyme, it basically just cuts at the ribobase. It needs a perfect match, and it deblocks the primers, allowing Taq polymerase to extend. The secret to this being very effective for PCR is that our RNase H2 is a thermophile, so it only gets going at 60 C. So at the temperature where PCR works. So all your primers are in the pool, they anneal to their targets, and they can then get deblocked and extend. So this is very useful for highly multiplex PCR. I'm not going to go into the details here, but if you're doing on and off target editing and you need to, assess in a single PCR dozens of amplicons, this is a technology to use. But we use this for everything, for on target assessment as well as off target. And so we can design the primers automatically for you that flank your cut sites of interest, and it's basically just two PCR. PCR one is to create the RH amplicon, and then PCR two is basically just a regular PCR to introduce the Illumina adapter. So all this in the end creates an Illumina sequencing library. And this is the tool that's on the website. We just need to have information about your target of interest. So essentially you need a bed file. A bed file contains chromosomal location, the start and end of the spacer for the guide RNA. This 0 here doesn't really do anything. It's just an insertion into the columns. But we do like to have strand information, right? Is the guide RNA facing this way or is it facing that way? For the design of primers, not something that's necessarily needed, but for the analysis part, which is what we also enable, you need to know if it's facing this way or this way, because the cut can happen here or here. So we need to know which window we're looking at. So I always tell researchers, provide strand information. But the tool is very simple to use. You select the application. In your case, it's genome editing. You provide the coordinates like I have here, species, insert everything else. You keep it default, and we design the primers, and they can come to you in plates or in pools, in tubes that you can then use to do the two PCRs. PCR1 to create the initial amplicon, PCR2 to add in the adapters. Really, the most important, I think, part of this workflow is not necessarily the PCR part to me, but it's the analysis part. Everybody can generate PCR amplicons. Most of us can generate a lot of NGS data. Where most of us struggle, including me, is how do I take this sequencing data and make something meaningful out of it, something that is informative. And we found that a lot of researchers struggle with analyzing NGS data. So what this system offers you is actually access to an analysis tool. When you order the library kit and the primers, you get access to a tool that allows you to just take your Illumina FASTQ files and just drag and drop into our tool. It's really that easy. You go from your computer and you just drag them into the tool. You provide information about the sample, so the guide RNA, the orientation of the guide, the amplicon that you're targeting, all the little details that we would need, and the tool can provide all the analysis you need from the kind of or the overall editing rates, the types of editings that happen. And it can go down to every single read, and you can do this one target at a time. Or as I mentioned, if you're doing on and off target editing, a single sample, we can design amplicon pools that can potentially do hundreds of PCRs or amplicons in a single PCR reaction, All that will be done and analyzed in a single analysis. So it's very useful. It has really accelerated the adoption of NGS as an analysis tool. Just because people don't have to think about how to analyze it, you don't have to worry about a bioinformatician in your department to analyze your data. Every researcher, can analyze their NGS data seamlessly and easily. And this is just a kind of high-level overview how that entire CRISPR system or the analysis system actually works. So we have the design tools I mentioned that can do all sorts of things for you, from singleplex analysis to highly multiplex analysis. We have a library prep kit which has reagents for PCR and PCR2, so the RH-PCR plus the PCR that introduces the adapters. You can sequence your data anywhere and then come back to IDT with your FASTQ files to do the analysis. And lastly, we've gone one step further at IDT. So a lot of researchers can do all this analysis on their own. But for somebody that's looking to take a target with CRISPR into a therapeutic setting, there's one more thing they need to do. They need to demonstrate the safety, right? They need to make sure that this is not unsafe for patients. And how do you do that? There's a few ways that you can potentially think about assessing the safety profile. But the way we do it at IDT is we use an improved guide-seq approach. So guide-seq, if you're not familiar, you introduce a double-stranded tag with your guide and Cas9 into a cell of interest, and that tag integrates wherever there's a double-stranded break. You can then shear DNA, the sequence of the tag, the adapters, and you can start identifying where the potential off-target sites are. So we use an improved guide-seq approach to nominate potential off-targets. And we do this at IDT in a very promiscuous system that will generate as many off-target events as possible to give you increased sensitivity. So we have this, what's called the off-target nomination workflow. We also do verification. So we'll provide the nominated off-target list to our customers. They'll do the editing in their clinically relevant cell types, and they'll send us back samples, and we'll do NGS sequencing analysis, looking at very high depth of coverage, analyzing editing rates, translocation rates, all the details that you would need to have a report that you can file into a submission. In the US, it would be an IND that you would submit to the FDA. We have a lot of examples where we consulted and we guide customers through this process. But this is a service we offer. So as an example, you have a guide RNA that you're interested in, that you want to put forward into the clinic. You can come to IDT. We first have a consultation to understand what you're trying to do. Once we align on all the details, we basically just need to know what your guide RNA sequence is. We will then make that IDT, run it through the workflow, nominate all the off-target sites, and then we'll have another consultation call with you with all the reports that we generated. You then, as a researcher, can pick and choose which targets you may want to analyze in your cells of interest. We will do the editing, and you let us know, this is the panel I want you to build. This is the panel that I want to be my verification. And we'll do the sequencing for you, and again, provide a report that can be used in your internal technical consultation, as well as any sort of regulatory body submissions. So this is our, what we call CRISPR on and off target edit analysis services. It's not a product you can order on the website. There's information there. It's almost always done through consultation. And you start with me for that, and then I can connect you with the rest of the teams at IDT that actually do the bench work to generate all the data. And lastly, functional genomics is another portfolio at IDT. So we do a lot of CRISPR genome editing, a lot of tools, a lot of innovation, but I don't want to lose sight that CRISPR sometimes is not always going to be the answer to every problem you have. There are genes that you just cannot knock out, because if you do, the cells don't live or the animal doesn't live. So some things are just not, I think, amenable to CRISPR, right? We have tools that allow you to do gene knockdown, whether it's through antisense technologies or RNAi. So just keep that in mind. We can make ASOs, lots of different chemistry options. We also design them for you. So there's a few different options for ASOs. Typically for gene knockdown, you would use a gapmer design that takes advantage of RNase H1. We have tools available for that, and we can make this. As a matter of fact, IDT was founded by a professor who has the first patent on antisense technology. So we've been making ASOs and working on antisense technologies much longer than CRISPR. So keep that in mind if you need it. And likewise for RNAi, if you need to do gene knockdown using sRNA technologies, we have both a design tool as well as all the reagents and support that will come with that. So with that, I'm going to thank you for your time. Gracias por su tiempo y atención. Yes. In plants, I don't have evidence for that. We do have researchers that use both our Cas9 enzyme, and then very interesting, the Cas12a enzyme is actually much more widely used in plants from our portfolio. So the enzymes work well, but the HDR tools, I have not had anybody. I would say the HDR enhancer protein is evolved against a human protein, so probably not a good idea. The small molecule, I don't have anybody that I know that has successfully done that type of application and said that there's a boost in HDR rates there. So unfortunately, no. But there's a couple of papers that you can look into where IDT's LB Cas12a. So we have two different Cas12a enzymes. One is AS Cas12a, which I show was used by Editas. There's another one that I didn't go into detail, but it's the LB Cas12a. That one is particularly useful in plants because it works much better at a lower temperature. So we have lots of researchers that have deployed this as an RMP using particle bombardment or using PEG media transfection to transfect embryos. So that works well. If you want those papers, I can send them to you. It was work that was done in maize and rice and tomatoes and potatoes, things like that. But generally, RMP is slowly gaining traction in the plant world, but it's still a very agrobacterium-dominated editing. That's yeah. It's easy to implement, but if you need to see how those tools are deployed, yeah, there are people that do, but HDR is definitely one on not something that I've seen used. And then you had another question. Uh, mhm.
And the synthesis engineers say, well, this is not possible to synthesize that because of the repetition of the promoter, etc.
Yeah.
And it was something like 5 years ago. I want just to know if it's still a problem in your company to synthesize just the cassette of the text.
Yeah. I would need to see the sequence to give you like a more informed answer, but I can tell you that it's gotten much better, much better. So your issue is you want to clone in a cassette that can express Cas9 or Cas12a, and so there's common sequences that are just repetitive. Yeah, 5 years ago, I can see that being a problem. How big was the construct? Monkey. Okay, so you had maybe like
something like
5, 6 repeats. Yeah. So there are two ways that we have changed over the last 5 years. One is to just being able to accept more complex sequences. And there are two ways you can potentially do that. One, we've improved the manufacturing, so that's one. So we can just take on more. Repeats are always hard, right? Like we can take more GC content and work around that, but repeats tend to be more challenging because we're using oligos to assemble. So same sequences coming back together creates a lot of issues. However, there are increasing complexities that we can accept depending on the product type. So as an example, a linear double-stranded fragment has much more stringent criteria than making something a plasmid. So that's the same sequences that we could take in a plasmid can be more challenging to make than in a linear fragment. The reason for that is that we can make a very poor quality double-stranded fragment that might have what you're looking for, we can't sell it to you because the quality is just not good enough. But we can clone it and just look for clones that are perfect. So it allows us to take more challenging sequences in a plasmid format. The other one is we've partnered with a company, Anza Biotechnologies, which you'll hear a little bit from Riley. It's the latest collaboration in industry. They take on much more complex sequences. So what might be challenging for IDT in many cases because lots of GCs or repeats, they have a different way of synthesizing the actual starting material. Where IDT makes ultramar oligos, usually 120 nucleotides, and we overlap them to create genes. They do enzymatic synthesis, so their oligos can be 1,000 base pairs long, in which case they can overcome a lot of complexities that shorter oligos can overcome. So I would say let's take a look at it again. I can tell you definitely what was possible 5 years ago is very different today. Very, very different. So I would love to take that on. Send us the sequence and see what our team has to say. I think it'd be a good test run.
If with the new technology I could make more multiplex.
Yeah, I think this is a really good test case for what we're trying to do is to say yes to everything. So
thank you very much.
Yeah, you're welcome. Okay. Is Rachel not feeling well?
Thanks, Ram, for your presentation. Now we are proceeding to the next presentation, which will be led by Riley Engstrom, who is our channel partner manager for Latin America. He has experience with the NGS portfolio, and he will talk about this. And he's been with IDT for 5 years. He has a graduate studies. He has a graduate degree in biochemistry. So just to introduce your talk, there's a question on the transmission on YouTube that maybe you might want to talk.
Question
And it
for
is if IDT has neurodegenerative panels or other disease panels for NGS and library preparation, which
MRD.
is
that what you said?
Yes.
Sorry.
Hold on. Here is the question. Do you have libraries for specific applications like for neurodegeneration, brain cancer, or immune cells?
Is that like off-the-shelf libraries? No. And is that a question for Brandmore or?
I think it is
Because I don't see NGS in that. That could be cloning, or because we can do custom synthesis libraries, which I will touch a little bit on too as well.
Okay.
Whether it's for cloning, CRISPR libraries.
Well, thank you, Riley. Welcome.
Okay, thank you. All right. So as David said, I am the Latin American Channel Partner Manager. So I started covering all of Latin America at the beginning of this year. So if there is not a distributor such as Uniparts, I do also sell directly to other countries. Perfect example, and we'll talk about for your situation when we hit synthetic biology too, is that there is a shrimp farm down in Bolivia that I've been working with, and we have been doing some complex clonal genes as well. I will start with next generation sequencing though. When I first came to IDT, I had a very big interest and then shortly became the next generation sales specialist for the Eastern Americas and all of Canada for a short period of time. Then IDT said I get to learn everything. So I will start with this. So when you think of next generation sequencing and IDT, pretty much everything post-extraction for all workflows we can help with. Even if we don't have a complete workflow for it, there's a lot of various aspects that we can, especially adapters and primers. But we will talk a little bit about hype capture today for the most part. So this is kind of just looking at the various XGen workflows that we have. As you can see, whole genome sequencing, whole exome, hype capture, amplicon sequencing, which we can do custom designs for those as well, which I'll talk a little bit on. Whole genome bisulfite, targeted methylation, whole transcriptome, and targeted RNA sequencing. We do not have single cell at this moment. We can help generate libraries for that, and of course, adapters as well. So if you do ever have questions on that or get stuck, if we are able to help, we can help just troubleshoot pilot ideas back and forth. Okay, so going into our library prep, these are the various DNA kits that we have. So there is the DNA library prep, EZ, enzymatic fragmentation, MC, mechanical shearing, the cfDNA and FFPE, ssDNA, and methyl-seq. Those will all be mechanical shearing, typically through a covirus instrument. A couple of the key features here is looking at the input value. The cfDNA and FFPEs, or cell-free DNA, often called, inputs are as low as 1 nanogram to 250 nanograms. Our single-stranded DNA doesn't have to be single-stranded, can be double-stranded, and low input can go down to 10 picograms. Those two kits are typically good for any more difficult, challenging samples or lower input values. And then, of course, our methyl-seq. Also, please feel free to stop me at any time if you do have questions. This is just looking at the workflow through end repair, 2 ligation steps, and PCR for our cfDNA and FFPE kit. These do use unique molecular indexes, so these are full-length adapters that are attached to this kit. This is great for larger library samples, larger sample volumes, and needing that specificity between each of those cell cultures. And then our RNA library prep kit and our broad range RNA library prep kit. The biggest difference between these two is the hours and the input level. So total RNA versus mRNA, and then similar to the ssDNA and cfDNA kit, if you have damaged or low input values, that broad range is good for you. It's just an extra hour. Up to 1,536 libraries is question? No? Okay. We do have up to 1536 UDIs off the shelf for these. If you do want more, we can. I have worked with people that have requested up to 8,000 or 9,000 UDIs at a time. We do make all those in-house. We do have those sequences available to you as well. This is, again, looking at the workflow, showing the additional time that the broad range RNA kit takes with that extra ligation step, and then the indexing PCR at the end. So adapters and primers, as I stated before, UDIs, UDI UMIs, we do have combinatorial dual indexes still. We generally recommend moving away from that if you can, as it does increase the chance for index hopping, crosstalk, and then we do have single indexes available as well. This is just looking at the various differences. I can share these slides with everybody afterwards as well. And then our hybridization capture. So we have all these various or more commonly utilized situations where people are using our hype capture panels, whether it's off the shelf or custom. This is the general workflow here. So obviously you're generating your own libraries versus amplicon sequencing. You have the hide and wash step. And then here is the list of our most commonly used off-the-shelf panels. We do also have off-the-shelf amplicon sequencing panels. The nice thing about this, though, is the custom panels that we do offer. Say you have a unique set of targets, you don't necessarily have the time to design, or you just want an extra set of eyes to help with your design process. We have a group of about half a dozen people that are our design specialists. If you submit your bed or FASTA files, we will take a look at that, and we can give you out a list of probes or primers that we have designed for you. We will mark them as good, bad, ugly. Bad and ugly obviously have a higher off-target analysis chance, but we can have that conversation, decide. Also, if you don't know what necessarily is going to be the best for your project, whether it's amplicon sequencing or hype capture, we can have that conversation with you as well. And like I said, this is free of charge. There is no commitment to buy. Like I said, if you ever have something that's difficult or you're new to this, you just want to talk it through with somebody that does this every day, more than happy to set up that call with you guys and Uniparts, we can talk this through, make sure that you're feeling more confident and ready to go with your specific project. This is looking at our high V3 reagents, so the magnetic beads and buffer. We have done quite a bit of improvements here. As you can see, the hot buffer handling is completely removed here, and the minimum hide time is 1 hour, and minimum input went from 500 nanograms to 100 nanogram. This is looking at it in comparison with some of the more typically used hide and wash buffers that are out there on the market. I'm sure you're familiar with some of these. If you do have any questions on this specifically, please feel free to come up to me afterwards. And that's NGS. Before I hop into custom oligos, qPCR, and SynBio, was there any more specific questions on NGS that anybody had? Even if it wasn't on those slides that I could maybe help with. No? Okay. Okay. Now, I am very well aware that most people in the world, if they know of IDT, they know us for primers. So I don't want to go through specifically our oligos. I want to just highlight a couple of the new things that we have and maybe a couple of capabilities and tools that you may not have noticed in the past, or a lot of people that I have come across were unaware of. First thing is our modifications. If you ever play around on our webpage and you go to custom DNA oligos, if you go down before, there's the modifications tab. You can click on that and it'll give you various additional tabs that you can click on, whether it's linkers, phosphorates, etc. There's probably a few hundred of those on our website. If you're looking for a very unique type of modification to put on an oligo, but you're not seeing it, we can review those, and we typically can synthesize those ourselves. We have done a couple thousand other off catalog, as we talk about, synthesis requests for people with unique modifications. It's very rare that IDT is unable to synthesize something. Bran hinted at it a little bit earlier. I like to think of IDT as pretty much, if you can think of something with DNA and RNA, IDT can usually review that, and we can most of the time commit to making something that you're looking for. This is going over our purification. So obviously most oligos are standard desalt, HPLC, page, dual HPLC. And then going back to the fact that I said we can customize oligos in most fashions, we can also deliver them in most fashions. So whether you want aliquots, double-stranded, multiple plates, multiple tubes, unique plates, we can review those. We've taken in plates from other plate manufacturers and shipped out oligos in those for customers before. So like I said, there's really, if you can go about as custom as you want when it comes to working with IDT. And then our primer and probe assays. So for qPCR, whether it's cyber green or TACMAN based or just standard PCR, obviously the primer assays are 500 reactions, assuming in a quantity of 5 nanomole. And then our primetime qPCR, the TACMAN assays, the minis are 100 reactions, standard is 500, and then the XLs are 2,500 reactions. This is looking at the double quench probes, so just reducing background noise. There is that option if you do want it. If you're ever having issues with your readouts from your qPCR runs, please feel free to reach out. Always happy to go through a data dive. If there's anything we can suggest or help you troubleshoot with, we can do that. Same thing for affinity plus probes, locked nucleic acids. If you need to increase the melting temperature point by a few degrees Celsius. And then there's various tools on our website as well. There's the Primer Quest tool, and then there's the Primetime Pre-Designed qPCR Assays tool. So Primetime Pre-Designed qPCR Assays tool, if you have your target in mind and you put that into that tool specifically, it will give you a readout of options, and it'll be given a score. for these primer probe combinations. If you ever see a green check mark by those, that has either been validated by somebody in the field or it has been bioinformatically validated on our end. The primer quest tool, same scenario, just won't get the green check mark with that, but gives you the option for cyber green assays and standard PCR. Oligo analyzer tool, looking at primer dimer formations, blanking on the other term, and then our multiplex dye selection tool. So this is great for multiplexing with qPCR. There is a large drop-down menu with a lot of options showing various qPCR and ddPCR instruments. It will show you for each well what the recommended fluorophore wavelength there is, what fluorophores we have that will go for that, and what alternatives you can choose as well. This is showing where you can find those on our webpage. So right on the top, tools there, qPCR assay, and then oligo analyzer, UNA fold. And then one of my favorites to show, especially in academia, if you ever have undergrads working in your lab, they're learning to pipette, that resuspension and dilution calculator can help save a lot of time. We do have custom design services for qPCR and ddPCR as well. So you don't have the time, you just want somebody else to do it for you. There is a small fee involved with this, but we can design for your targets. The design team here has a little bit more than what you would see in terms of our online tools, a little bit more bioinformatics on their end. But here are some considerations, primarily for dqPCR, ddPCR. These are some more inhibitors, things to consider. And then recently, we have developed our primetime pathogen panels. So there is 211 targets on these. These are off the shelf. All of them are able to be multiplex except for the North American tick panel. As you can see, there's urinary, AMR, respiratory, STI, GI, wound, sepsis. A lot of people have been using these in the States for research and other purposes upon creating LDTs, etc. Is something that you can consider if you're doing it. I have seen this being used in research as well for some veterinarian populations. It's very dependent though, as these are human designed panels. So if you ever have questions on that or think something might be worth inquiring about, we can cross-reference for you and let you know if it would work. This is just showing how to order it online, the available fluorophores. One thing I do want to note too with each of these, let's say you're interested in the respiratory panel, you do not need to grab all 32 targets there. You can customize this as much as you want. You could honestly customize all this together and create one panel and say you wanted 4 of urinary, 4 of respiratory, 4 of STI, etc. You can create your own. And then we can also go ahead and if you have any specific pathogens of interest that are not on this list at all, you can add those to those. So again, going back to the customization, we can provide about as much as you want. We do have master mix for TACMAN-based assays. A lot of people are unaware of IDT as making master mix as well. The most beneficial one, especially for samples that are, again, crude, low input damage, is our primetime 4X broad range master mix. We have tested this in-house up to a 5-plex assay there. It does come on dry ice, so it is a little bit pricier on your end. If it is ever something that you guys would like to try out, please reach out to Uniparts and they can reach out to me. I'm more than happy to put good working solutions in your guys' hand for you to test out. And this is kind of looking at the variations of there. So the broad range is a 4X. The one-step RT-qPCR standard master mix is a 2X along with the gene expression master mix. The gene expression master mix is very nice because it comes lyophilized, so it can ship without dry ice. It is much more budget friendly if that is something that you need to take into consideration. Any questions regarding oligos and qPCR or ddPCR? Okay. Then synthetic biology. So here's the list of various fragments that we can make for you. There's the E-block, so if you're doing antibody discovery, don't necessarily need the highest of quality. We synthesize these the same way as D-blocks. There's just a step less of quality assurances more there, and we can churn out much faster and at a cheaper rate these plates for you. It does come with a minimum of 24 fragments that are needed per plate. And then our standard G blocks, which are often used for qPCR controls, gene constructs, and then our hi-fi fragments, which are the G blocks gene fragments, just with NGS verification. So you are getting exactly what you requested in terms of the sequences. There's no mismatches or incorrect base pairs. And then our gene synthesis. So we have a list of about 10 vectors off the shelf that are not an additional cost to you. You just pay for the gene insert there in those vectors. If you do have a vector that is not listed on there that you do want, we do have a custom vector onboarding service. There's normally a one-time $500 USD fee. Right now, there's an ongoing promotion until, I believe, June 30th of next year or July 30th of next year, where it's $500 off. So if you have a more unique vector, you can onboard that for free. You're just paying for the gene fragment. And then that vector is on your account forever. So you don't have to pay any recurring charges once it's already on there. So something I like to point out. And then our rapid genes, these are done in the off-the-shelf vectors as well. These are NGS verified gene fragments though, and those are at a cheaper rate and shipped out in larger quantities. So those will come in plates. And you can customize gene in A1, gene fragment X with vector or plasmid Y, and then A and B and then X, etc. This is just kind of looking over our synthetic biology portfolio in the various areas that we often see it. And then, again, going back to more tools on our webpage. So there's the complexity checker, codon optimization, check on your order status. And then our IDT-ANSA partnership. So this is for fragments such as yours that were shorter, said not possible. This team will review. We can go up to 50,000 base pairs with them. This is a little bit of the process online, how you see that denied high complexity score. And then there's what it looks like now. If you go to do this and there's a high complexity score, we can have our friends at ANSA take a look at this. In coordination with our partnership with them, we decided to go through our SynBio Custom Solutions program. And that's what I was explaining to you about the customer that we had in Bolivia that has some more complex gene fragments. If you have an idea or a fragment, you're getting errors or you're being told it won't work, this team will come together. We will talk with you about your project, what your overall end goal is, and see what we can do to help get you there. Basically, you'd fill out the SCS form that was on that webpage. If you get that high complexity, you can then converse with me and our friends here at Uniparts. We'll do the free consulting, budgetary estimate, statement of work, manufacturing, and shipping. On top of that, too, we have a computational library design as well. So I hope that answers the online question that was there earlier. If not, please reach out. More than happy to help there. And that's all. Any questions for me? Anything that I didn't cover that you guys wish I would have or that I could help with? I do have my business cards down here. Please feel free to grab them, reach out. Like I said, I've worked with IDT. I try to provide good solutions to researchers. I was in academia myself for the better part of a decade. If I can ever be of assistance, I am more than happy to, whether that is financial, billing questions, or technical inquiries in terms of what we're capable of providing. Please reach out. I'm always happy to help.
Thank you. So it's not, again, part of the qPCR or what you present, but do you have any expertise on oligo paint
Oligo.
and kind of fish probes, but
W-
based on oligos? Like
So
primates?
we can't help support because we have not done any internal research on our end when it comes to fish assays, but we have supplied fish probes for customers for a while. I've worked with a few customers directly on that, but we don't necessarily have tools or scientific support services there. Okay, all right. Thank you very much, everybody. I appreciate your time.
Muchas gracias por haber venido. Ah, sorry. Okay. Bueno, a los que nos estaban viendo por internet, pues muchas gracias por haber asistido. Les brindamos, como dijeron, las puertas abiertas para que se acerquen a nosotros para algún proyecto en específico. Entonces se pueden acercar a Uniports y con mucho gusto los canalizamos con IDT. Y manténganse abiertos a nuestras redes sociales porque ahí estamos publicando webinars. Por ejemplo, la siguiente semana va a haber uno sobre cultivo celular por uno de nuestros asesores científicos. ¿Vale? Que tengan excelente día.